in some sort of, if you have multiple models involved in some sort of cell biological process, it's not very practical to use molecular dynamics because usually the time scale here is microseconds at the most, and the spatial scale is on the scale of individual molecules. And then at the other end of the spectrum, you have the kinds of modeling we've been doing so far with Copasi and virtual cell, where the scale is usually on the level of entire cells. In this case, the molecules that are involved are modeled as infinitesimally small points. We're not really taking into account the shape and the size of the molecules and how they might fit together, how they might interact. But the time scales and the spatial scales are nice if we need to model very complicated reaction networks. We can do it for seconds or minutes or hours, depending on the kinetics that we're modeling. And of course, the spatial scales are on the size of a cell, if not bigger, even tissue. But what if we do have to worry about the shape of the molecules and how they fit together? We can't do that with virtual cell or Copasi. So SpringSalad is in this intermediate range where we're dealing with time scales of maybe a second, not too much more than that, and spatial scales of, say, a few hundred nanometers, much bigger than the size of a single molecule, to try to encompass how many molecules fit together. And the way we do that is using coarse graining to model the molecules, which do now take up space. But that we're modeling them as a series of spheres. So let me go on to the next slide so you can see that a little bit more clearly. The paper that was published in 2016 is probably the best source if you want to go into this in detail. It has a very extensive appendix and supplementary material that includes a manual. But the manual is also available online through our website. So through the vcell.org website, there's a tab called SpringSalad, and you can get all the information there. And you can also download SpringSalad there if you haven't already done so, if you need to do that. The way it works is we model molecules as a series of connected spheres. And the spheres do take up space. They are volumes in space, in the 3D space. And they're connected to each other by stiff springs. And the advantages of this is that it actually does model how molecules can fit together, at least in a very coarse way. It does exclude volume. You take into account steric hindrance. It captures the dynamics based on measured, hopefully measured, reaction rates. So it's not like molecular dynamics where you're dealing with potentials to try to model the molecular dynamics. Here we're dealing with the same kinds of inputs that we use in VCell or Copasi, reaction rates and diffusion coefficients. Those are translated into the microscopic parameters, the microscopic on rates, the microscopic off rates, the microscopic Brownian dynamics that's required for these models. So the software does that automatically. So let's take a little closer look at how it does this. Each of these spheres that you model in the SpringSalad molecules is semi-independent. It's independent in the sense that you assign it a diffusion coefficient, and from that diffusion coefficient, the software will generate a series of random forces that impinge on the sphere to make it move in different directions. The forces are in random directions and random magnitudes, but the envelope of those random magnitudes correspond to the diffusion coefficient, or come down to the diffusion coefficient that you've assigned it in the inputs. Okay, so the diffusion coefficients are translated into these random forces that make the individual spheres move in different directions, but in proportion to that diffusion coefficient. And the spheres are connected to each other within a molecule by stiff springs. And the reason that that's done is that the stiff springs are allowed to transmit the forces. They don't really stretch or contract very perceptibly within a simulation, but they're using the springs allows the software to transmit those random forces to neighboring spheres. And so the entire molecule can ultimately diffuse based on the random forces that each individual sphere experiences. Okay. And this is just an example of how a few molecules might be translated into these coarse-grained collection of spheres. This is, I guess, a histone being translated into two spheres, or obviously it goes on much further, but these are little clumps of DNA and protein that are connected by short strands of DNA, and this would be a polymer. This is, I think, hemoglobin or myosin, maybe, which is translated into four very closely packed spheres. And this is a tubulin molecule with a GFP attached to it. So this is how you would model that. You can model it more, it doesn't have to be this coarse, but this is just sort of an idea of how these models are developed. And here's a screenshot of automatic model creation from a PDB file, and I'll try to demonstrate that before the end of the talk. I mentioned that we do model biomolecular reactions based on rates. This is just to show you how that's done. This is the on rate that's input, and from the on rate, the software calculates the probability of a reaction between two spheres that might react between two sites based on a probability. So, you know, the math is complicated, but if you're interested in how that's done, you can look at that paper that I cited before. So let me give you a quick example, and I'll show you this, a version of this in the software itself. This is an example of a three-component system. The molecule nephrin, which is bound to a membrane. So there's a little anchor here on the membrane, and it has three binding sites that are phosphorylated tyrosines. Those phosphorylated tyrosines are in red here, and they can bind to an SH2 domain on this molecule called NCK. And the NCK also has three SH3 domains, which are in pink here, and they can bind to any of the green guys in this molecule called NWASP. These green balls are binding sites representing proline-rich motifs, PRM motifs. So PRMs can bind to the SH3s, and the SH2 can bind to the phosphotyrosines. And you can see this is seems like a simple system, but the number of ways that these molecules can bind together can be quite complex. And this is simulated in a cubic reaction volume, 100 nanometers on a side. One surface of the reaction volume is a membrane, which is where the nephrin is anchored. And we're modeling it with a total of 36 molecules, 6 nephrins. In this simulation, I'll show you 20 NCKs and 10 NWASP. And this is what the simulation starts off looking at. We have everything all stretched out, but at the end of the 100 millisecond simulation, things start collecting and they end up on the membrane. And I have a little movie of this here, but I'll show it to you in the software as well. So this is the kind of output that you can get from SpringSalad. And you can see at the end of the movie, there are a few molecules that are left in, a few complexes that are left in the cytosol, but most of them are now associated with the membrane. Okay, so now I wanted to give a quick demo of the software itself. This is actually a simpler version of that model where I don't have any intermediate binding sites. So the NCK model is just it should be, there's a third ball out here. I have to change the size of the canvas. Yeah, now you can see the third ball. So this is just out in space. And in this model, well, we can change the number of molecules when we do simulations, but this is the definition of this. And as you can see, you can, if I click on here, this is the SH2. You can give it a radius. You can assign it to a particular position, and you can assign the links between the different sites. So we have various links here, and the length of those links is assigned as 4.5 nanometers. If I look at NWASP, find it, here's NWASP. This is just an N site that isn't involved, and we have 5 PRM domains. So there's these are the PRM domains, 5 of them, and there's one site that we're calling BCA, which we might use for something else later on. And then nephrin has only 3 active sites. They're all tyrosines, phosphotyrosines, and then it's anchored to the membrane. This yellow line here represents the membrane, and it's anchored by this site here, which we also have to give a diffusion coefficient to. So here we've given the anchor site a diffusion coefficient of 0.1 micrometer squared per second, which is sort of what the diffusion coefficient of a membrane protein would be. But the phosphotyrosines actually have diffusion coefficients of 10. So even though they have faster diffusion coefficients, they're constrained because they're linked to the membrane. So they can flop around in the cytosol very well, but just adjacent to the membrane. They're tethered to the membrane and don't diffuse very much in the plane of the membrane, just diffuse around in that confined space near the membrane. Okay. And then we have all of these different types of reactions that we can define. Let me just show you one binding reaction, for example. Here's the SH2 domain on NCK binding to one of the phosphotyrosines on nephrin, but it depends on the phosphotyrosine being in the phosphorylated state. So we have two states for that tyrosine. So if you remember Michael Blinoff's discussion of rule-based modeling yesterday, this is basically the same as rule-based modeling. And in fact, I won't have time to show this to you, but in our development version of VCell, we are in the process of adding SpringSalad as a new application type. Currently in VCell, there's ODEs, PDEs, non-spatial stochastic, spatial stochastic, NFSIM. All of those are different application types, and you can have many of each of those. We are in the process of adding SpringSalad as a sixth application type for a new kind of simulation. And it'll fit fairly easily based on the fact that it's using rule-based modeling. Okay. How much time do I have? Good. Let me show you a simulation result. So if I go to Tools and bring up the Simulation Map Manager, here it is. I have two simulations that have been run. One is a simulation with 10 runs total. I hope you can read this. A second simulation just has one run. So let me start with the 10-run simulation in case we don't have time. This was run, if I remember correctly, for 100 milliseconds. Let's look at a 3D snapshot. So again, at the time 0, everything is sort of randomly placed and stretched out. And you can see the nephrins here that are anchored to the membrane, the cyan molecules. I think there are, did I say 5 of them? I think there are 5 of them in this simulation. And then if I play it, you'll see exactly what you saw in the slides. These things are bouncing around, and eventually a lot of them end up on the membrane. So this is just 10 milliseconds, but you can see that many of them are sitting on the membrane. Right now, there seems to be just one guy that's hanging out in the cytosol. The other thing that you're seeing is that within the membrane, they seem to be highly interconnected. There's one nephrin over here that seems to be holding on to a couple of complexes of NWASP and NIC. And then there's this larger complex of totally interconnected nephrin and NWASP. So how can we really analyze what's going on here? Well, we have some very extensive data analysis tools in the software. So let's start off with this simple one, molecule count. If I hit bound NIC, this shows that at time 0, there's 0 bound NIC, and then it gives you a time course. And you could, of course, copy this into a spreadsheet and create a plot and do whatever you want with it. It's a little bigger. Yeah, this is the standard deviation of that. And you can do that for, this is being done for all of the data for those 10 runs. So this is averaging all of that data. You can also look at histograms and etc. So for bound nick, this is the histogram for all of those runs. But let's look at another data class. Let's look at cluster statistics. So under cluster statistics, let's start with the average cluster statistics, and this is the mean for all of the runs. We have different kinds of statistics that we've calculated. This is the average cluster size over those runs. So you see as time goes on, the cluster size gets bigger and bigger, the average cluster size. This is the mean average cluster occupancy. And the definition of this is up here. I don't have time to explain that, what that means, but it's essentially a better metric for the average size of a cluster than just the average cluster size. And we can also look at histograms. Looking at, this is the mean, but let's look at a time, this is a time 0, so there's not much going on. But let's look at time 10. You can see that we have the frequency of each cluster size. That's the average. And we also can tell what the composition of those clusters are. So at time 10 milliseconds, you can see that there are 27 clusters that are quite large with a 15 nicks, 7 NWASPs, and 5 nephrins. So you can get all of this detailed information out of this. And you can also look at individual runs. So in the movie, I showed you run 0, and you can see that in run 0 at 10 milliseconds, we indeed only have 3 clusters. One of them doesn't have any nephrin. That's why it was out in the cytosol. And the other 2 have nephrins. One of them has just 1 nephrin, a bunch of nicks, and 2 NWASPs. The other one is a big clump with 4 nephrins, 12 nicks, and 6 NWASPs. So you can get detailed information about what's going on just by looking at the data that comes out of the software. And in my last 3 minutes, let's go back to the software, to the model, and I'm going to show you how we can load a PDB file. This is a PDB output format, a CIF format for NCK. I'm going to open it. Where is it? Oh, I'm sorry, I did the wrong thing. Add PDB. So now it's going to ask for the number of sites I want. Let's make it 4 sites because there's 1 SH3 and 1 SH2 and 3 SH3s. And it's going to ask for anchor sites, but or fixed size sites. I'm going to cancel that just to keep things simple. Where is it? This is what happens when you try to do a demo, huh? Okay, here it is. As you can see, it created 4 sites from this molecule. let's change the molecule to more easily seen style. Let's do a ribbon style. So now you can see the different domains as I rotate this. And if I click one of these, it will allow me to define it in the software. And that's how you can create a model directly from the PDB. So this site here looks like it's this terminal SH2 domain, and the other three sites are the connected SH3 domains, and we can define them in the software. All right, I'm going to stop here and see if there are one or two questions about SpringSalad. Any questions? Okay, I guess that was clear enough.
I have one question.
Sure.
So you gave the example of a phosphorylated site and non-phosphorylated site. Are you able to also use a rule to switch states within?
Oh yeah, of course. Yes.
Yeah. So when upon one binding event, then you could change the probability that another site becomes phosphorylated or dephosphorylated.
Right. So we have transition reactions. I don't have any defined here. I mistakenly opened up a new model instead of adding this to the previous model. Now let me open up the previous model. Okay, so transition reaction. So nephrine phosphorylated. So here's a reaction for going from unphosphorylated to phosphorylated, and you just assign it a rate. And obviously there could be a dephosphorylation reaction as well. And then we also have what's called allosteric reactions, where the rate of a reaction depends on the binding state of the system, of the molecule that's undergoing the reaction. So that, we have all of those possibilities. And those, yeah, so all of those reaction types are possible.
And then a follow-on question. If you have multiple tethered species, I'm just curious how, so the span of the membrane is relatively modest, I imagine, because of the computational limits. But if you have collisions between multiple tethered species in a relatively crowded environment, how does SpringSalad perform, or is there a limit to how crowded and how busy the membrane tethered component?
Well, I mean, if it, the molecules can't, the spheres can't pass through each other. So it will naturally account for the crowding. That's one of the strengths of the software. So if it's a crowded environment, that will be naturally dealt with properly in the simulation.
And are they confined? I guess, are the molecules, it looked like in the movie they're passing out of a boundary left and right, or there's some kind of a boundary rule, or are they confined like rigidly to the patch of membrane?
Yeah, they're reflecting boundary conditions.
Reflecting, yeah, that's, sorry.
So they can't pass through the boundaries.
Super, thank you.
You can make it bigger than 100 nanometers. In this particular model, it's 100 nanometers, but you can certainly go bigger than that. The simulation time actually doesn't depend on the size of the volume. It depends on the number of spheres that you use in your molecules. So yeah, you can use a much bigger volume if you want. Okay, I'll stop and hand it over to Pedro.